Hello everyone. Today we are going to be talking about the big topic of oxidative phosphorylation. Oxidative phosphorylation is the final process that we have been waiting up on until now in order to make 36 ATPs from one molecule of glucose. So firstly, let's sort of go over what we've discussed until now. Firstly, let me use a different marker. Firstly, this all happened from one molecule of glucose. That glucose was converted into two pyruvates from the process of glycolysis, and those two pyruvates will undergo pyruvate oxidation in order to make acetyl-CoA. That acetyl-CoA will then go within the Krebs cycle or TCA cycle in order to make electron carriers such as NADH or FADH. NADH or FADH. These are some electron carriers that the Krebs cycle produces from acetyl-CoA, and now these electron carriers go within the process of oxidative phosphorylation. It can also be referred to as the part where the electron carriers actually undergo this process can be referred to as the electron transport chain, or ETC. Now that we've looked at what we've done up till now, let's look at the whole process of oxidative phosphorylation in depth. Firstly, we need to understand where this whole process takes. Oxidative phosphorylation takes place within the mitochondria. So firstly, let's divide the mitochondria within three particular aspects or different regions. So firstly, there's the intermembrane, space. Then within the middle, right here, is where oxidative phosphorylation is going to occur. We're going to get into that. Down here is where mitochondrial matrix is present. So down here, there's mitochondrial matrix, and up here, there's the intermembrane space. So firstly, let's draw this out actually within a different color. Let's draw the complexes. There is a complex 1, then there is a complex 2, 3, 4, and there's the final process of ATP synthase and chemiosmosis that we'll get into. Now, there are two particular sort of, you can say, sub complexes that are, let me draw those in a different color or marker, sorry. Let me draw those in a different marker. So the first one is called, let's just put E right here. It's called coenzyme E. And after complex 3, there's another one called cytochrome C. Oh no, my apologies. This is coenzyme Q. Sorry for that, guys. And this process right here, this triangle, can be referred to as ATP synthase. ATP synthase. Or it can also be called, I don't have enough space here, but chemiosmosis. Those are two names. Sorry. Those are two names that can be referred to as the same thing, ATP synthase or chemiosmosis. So we need to understand that firstly, we have to separate this whole process of oxidative phosphorylation into two parts. So if we split the line here, the triangle part, if you just want to put it that way, is called ATP synthase or chemiosmosis that we talked about, and the rest of these complexes and mini complexes can be referred to just as the electron transport chain or ETC. However, the whole process itself is called oxidative phosphorylation. So firstly, let's go through the process. From earlier, from the Krebs cycle or TCA cycle, what did we find out? Let's use a different marker. Let's use a different one actually. We found out that NADH, firstly, NADH and other electron carriers, but just within complex 1, which we'll talk about in further later. NADH is an electron carrier that gets passed down from the Krebs cycle. NADH is then oxidized into NAD+, and that electron firstly goes into complex 1. Then as that complex 1 is supercharged, sorry, I forgot to mention one more thing. Let's draw a bunch of hydrogens down here. Let's use a different color for that. Sorry, guys, forget about everything I said. I made a huge sort of mistake a little bit. I forgot to mention one key detail. The main point of oxidative phosphorylation is firstly to make all of, if you want to put it, all of these hydrogens within the intermembrane space in order to create a proton gradient. So then all of those proton gradients go through ATP synthase again and go back into the mitochondrial matrix in order to make ATP. So let me repeat that. All of these hydrogens want to go from these complexes within the intermembrane space, make a huge proton gradient, and go back from chemiosmosis or ATP synthase and make ATP. So sorry about that confusion. Right now, you can say, the protons are right here. A lot of the protons are in the mitochondrial matrix, and there's a lot less within the intermembrane space. So firstly, like we were talking about before, NADH gets reduced to NAD+. NADH gets oxidized into NAD+. As NADH gets oxidized into NAD+, it loses the electron. That electron then goes into the first complex. Sorry about that, guys. Reduction means gaining an electron, and oxidizing means losing an electron. So NADH loses the electron, meaning it gets oxidized, and that electron goes into complex 1. Within complex 1, it becomes supercharged. So within this process, a lot of protons start to form. Let's just put those up there. So this is the first time a lot of these protons start going within the intermembrane space. Now, what do we do with the electron that supercharged complex 1? That electron then goes into coenzyme Q. This is sort of the intermediate that you can talk about. That electron goes into coenzyme Q. Now what happens? Now, let me just erase this for a second. Now, another process also occurs. Another electron carrier called FADH. It's called FADH. It gets reduced to FAD. Sorry guys, I keep messing up that. I truly apologize. I meant to say oxidized. When you oxidize something, you lose an electron. FADH is oxidized to FAD, losing that electron. So then that electron goes into complex 2. So let's put it right here. So that electron goes into complex 2 as FADH is oxidized. And as it is, as FADH is oxidized, that electron then goes into complex 2. And from complex 2, the thing about complex 2 is complex 2 cannot become supercharged. So from complex 2, that electron gets carried also within coenzyme Q. So from there, we need to understand two things. So in complex 1, NADH can only be oxidized, only NADH, the electron carrier NADH can be oxidized and used to supercharge complex 1, which allows a proton gradient to form in the intermembrane space. However, FADH only belongs to complex 2, which cannot be supercharged and allow protons to form within the intermembrane space, a proton gradient to form there. So that electron goes into coenzyme Q. What happens after? After that, once we understand that these extra electrons from complex 1 and complex 2 are within coenzyme Q, it then goes on to, those electrons then go on to complex 3. Guess what happens now? All of those electrons supercharge complex 3 once again and allow more hydrogens, or not hydrogens, you can say, protons, more proton gradients to form up there. And that electron again, let's see, those extra electrons then move on to cytochrome C right here. And cytochrome C acts as an intermediate. Then those electrons then go on to complex 4 again. Guess what happens again? Complex 4 gets supercharged, making more hydrogen. Sorry, not more hydrogens, more protons. More protons up there. So now, these electrons that were passed from cytochrome C to complex 4, where does it go now? Where does it exactly go? The final place it goes into, the final electron acceptor, you can say, is oxygen. So the final electron acceptor from complex 4 is oxygen. So those electrons are passed to oxygen, and oxygen is split into two separate ions. And after after those electrons allow oxygen to be split into two ions, protons are added from the mitochondrial matrix in order to make two water molecules, or H2O molecules. Two H2O molecules. That's the process from complex 4 to the final electron acceptor, which is oxygen. Oxygen then gets split into two ions, and within those two oxygen ions, protons are also added, making two water molecules. So now what? After we got that, why does it matter? Now, like we were talking about earlier, guys, you remember all of these proton gradients that have been forming up here? There is a lot less. We can actually remove these to show it. not just one. There would be more protons here, but this is just a situation where we're gonna represent that. Let's just draw one more just to show it. Anyways, all of these proton gradients up here are now gonna go through chemiosmosis from ATP synthase, and they're gonna go back into the mitochondrial matrix to make ATP. However, it's not that simple. Firstly, let's understand why our body wants to do that. Firstly, all these proton gradients, according to diffusion laws, our body always wants to go from an area of high concentration to low concentration, except if there's different factors, such as salt and all that. We're not going to get into that. We already talked about that in earlier videos. If you want to go check that out, you are welcome to. Those proton gradients go from ATP synthase to the mitochondrial matrix because they want to go from an area of high concentration to low concentration to achieve equilibrium. So within this area, ADPs, our body right here contains a lot of ADPs that are just waiting to make ATPs. So our body, from ADP, they get reduced to ATPs. There is a lot of ADPs present within the mitochondrial matrix here. They get reduced to ATPs. Reduced means they gain those protons. Anyways, ADP is reduced to ATP, which is incredible. And exactly 36 ATPs are made from one process or one breakdown of glucose. So this is a very crazy process. And this process would occur once again once there would be more electron carriers from the Krebs cycle, and the process would occur again. Some confusions within this process are going to be debunked real quick. Firstly, electron carriers such as NADH can only be found within complex 1. They can only go through the process of complex 1, and electron carriers such as FADH can only go through the process of complex 2. And now we know how oxidative phosphorylation works. And the other thing that we need to debunk, or if you guys didn't understand it still, is when all of these proton gradients go through ATP synthase, ADP here just wants to make ATP immediately so our body can use it for many biological processes. So ADP automatically reduces to ATP, and a lot of it is made. Thank you so much for watching this video regarding oxidative phosphorylation, and I hope all of you guys have a great, wonderful day. Keep continuing to watch more videos now, as now we know the whole process of how glucose is broken down. Now we can actually get into the whole process of how starvation and fasting actually works, because now we're going to talk about more complex topics which we need to understand in order to actually realize how much of a difference there is between fasting and actually starving. Bye guys, have a nice day.